Hello everyone, welcome to Chehal's Zoology Lectures. If you find any of the lecture interesting, please like and subscribe to this YouTube channel. Also hit the bell icon for further notifications. So today we'll talk about chordates. In chordates, we'll talk about similarity of chordates with higher non-chordate phyla. And the first point in this is bilateral symmetry. Now, many organisms from non-chordate phyla and almost all the organisms of chordate phylum, they possess bilateral symmetry. What is bilateral symmetry? If you pass the plane, sagittal plane, you cut the organism through sagittal plane. What is sagittal plane? That is a median longitudinal vertical section. You can see here in this picture. Here, this is sagittal plane. So this is median longitudinal along the length of the body and vertical section. So it will cut the body in two equal halves, which will be the mirror image of each other, left and right half. And it will be having anterior and posterior axis distinct, dorsal and ventral surfaces very much distinguishable. So such kind of symmetry is termed as bilateral symmetry. Now here you can see that all these insects which are part of Arthropoda phylum, they also exhibit bilateral symmetry. This butterfly, ladybird beetle, this bee, this beetle, they all have bilateral symmetry, and this is example of chordates. Also you can see dogs and cats, elephants, all kind of chordates, they possess bilateral symmetry. Bilateral symmetry gives polarity. That means animal moves with dorsal end upward and anterior end forward. Just imagine a cat or a dog walking. They have their dorsal end upward, their back upward, and their anterior end, that means mouth, forward. However, anterior end of chordates doesn't correspond to that of non-chordate. That means anterior end of chordate is different than that of the non-chordate because of the developmental dissimilarities. Now in this picture, you can clearly see here that chordates are deuterostomous organisms and non-chordates are protostomous organisms. Now what is this? Let me explain. Now here this is gastrula. Gastrula is having an opening which is termed as blastopore. Now in protostomous organism, this blastopore will develop into mouth. This orange structure, it will develop into mouth, that is blastopore. Okay, so this kind of organism, these kind of organism will be termed as protostomous organism. Protostomia. Proto means prior or first. Stoma means mouth. That is mouth is to develop first or prior than that of the anus. Anus will develop opposite to the mouth. Here you can see this brown structure, that is anus. So protostomous organisms are mostly non-chordate. Most of the non-chordates are protostomous except echinoderms, which are deuterostomous. So what are deuterostomous organisms? In which the blastopore, this orange structure, it will develop into anus, and opposite portion, this green portion opposite to blastopore, it will develop into mouth. Anus will develop first and then mouth will be developed. That's why these are termed as deuterostomia. Deutero means secondary or later on. Stoma means mouth. That means mouth will be developed later on in life. So such organisms are termed as deuterostomous. So anterior and posterior end of chordates and non-chordates, that doesn't correspond to each other. So next we'll talk about three germ layers. Now the organism which will possess three germ layers will be termed as triploblastic. Such organisms are termed as triploblastic, and three germ layers are ectoderm, mesoderm and endoderm. So all the chordates are triploblastic, and non-chordates above the phylum Coelentrates are triploblastic. That means organisms starting from platyhelminths, then annelids, then arthropods, then mollusks, and then echinoderms. These all are triploblastic. However, origin of mesoderm is different. How mesoderm is originated in chordates and non-chordates is different. How? You can see here, see in this picture here. Now, this yellow structure, this yellow portion is endoderm, yellow cells. These blue cells are ectoderm. Now, in between these two layers of cell, ectoderm and endoderm. At their junction, there is a group of cells which are shown here in red color. These are termed as mesoderm cells. So mesoderm cells will arise at the junction of ectoderm and endoderm. So such development is termed as schizophylic development. Okay, so that is development of coelom. We will talk about this later. So mesoderm will arise at the junction of ectoderm and endoderm in case of non-chordates. In case of chordates, it will arise from pouches of endoderm or enteron. Now this is archenteron, that means future gut. This portion will be in future it will become gut or alimentary canal. So this is termed as archenteron. In short form, it is also termed as enteron. So that is enterocoelic coelom and formation of enterocoelic coelom. So here we are talking about mesoderm. So this red portion, that means dorsal and lateral portion of this archenteron, it will give rise to mesoderm. So mesoderm will arise from endodermal cells. So this happens in the case of chordates. So non-chordate and chordates, they have different kind of development of mesoderm. So next, the third is organ system level of organization. Now all the chordates and non-chordates above phylum Coelenterata, that will be having organ system level of organization. That means now the basic level of organization is cellular level. That means each cell is different and will perform its own function. The next level is tissue level. That means some cells will group together, will make a tissue, and that tissue will perform a specific function. So there will be different tissues. So the body organization level will be tissue level. Cellular level is found in porifera and tissue level is found in Coelenterata. Now the next is organ system level. That means several different kind of tissues, they will form together a group and will function as a organ. And when certain organs, different organs, they will come together and form like organ system, and organ system, they will function together in coordination to make a body as a whole. Okay, so majority of chordates, all the chordates, they will possess organ system level, and non-chordates above coelenterate, that means platyhelminths onwards, they will be having organ system level. Next is cephalization. Cephalization means formation of head, that is termed as cephalization. Head has mouth, nervous system, nervous tissue, that means brain, and sense organs, eyes, nose, ears. So that is possessed by head. Now all chordates except urochordates, they will be having head. Urochordate, that means Headmania, Acidaceae, and Larvaceae, they will not possess any head. All the urochordates, they will be devoid of head, and many non-chordates, they possess head. In chordates, head will increase in size with higher groups. Now in this figure you can see that Cnidarians, they do not have head. Now the structures which are shown here, the groups which are shown here in orange color highlight. So these are the organisms which has cephalization. So Cnidarians means jellyfishes, they do not have head. Flatworms, platyhelminths, yes, they possess a head. Echinoderms, they do not have head. Vertebrates, all chordates, that means except urochordates. Cephalochordates also have head. Vertebrates, they possess a distinct head. Tunicates, that means these are urochordates. Acidaceae, class Acidaceae, urochordates, they do not have head. Nematodes, they have head. Arthropods, they also possess head. Porifera, sponges, they do not have head. So that was all about cephalization. Next is metamerism. Now what is metamerism? Metamerism is regular repetition of certain organs along anterior posterior axis of the body. That means certain organs will be repeated in sequence along the segments of the body, starting from anterior portion running towards the posterior portion. Now all the chordates, they possess metamerism, they show metamerism, and it is evident during their developmental stages, but it is masked in adult, it is hidden in adults. Metamerism is absent in non-chordates except annelids and arthropods. only these organism, these phylum shows true metamerism segmentation, you can say. Here is the example of annelid. You can see there is coelom in each segment. So coelom is here metamerically distributed. And also you have learned about nephridium that are also metamerically distributed in case of annelids. Now if you talk about chordates, you can see nerves are paired here and are distributed along this segment. So these are spinal nerves which are in pair in both sides, left and right, and repeated in every segment of the body. You can see there are several pair of spinal nerves. So that is an example of metamerism. Similarly, here in this lizard, you can see sensory ganglia. These are also repeated along with the segments. And spinal cord, in the center there is spinal cord. So the nerves which are arising, which are shown here in blue, that are spinal nerves. So these are again segmentally arranged. Okay. So metameres are basically somites which are arranged in segments. So that's why this is termed as metamerism, or you can say segmentation. So true segmentation is found in all the chordates, and annelids and arthropods in the non-chordates. Now next is appendages. Now what are appendages? Appendages are uh, organisms body parts which will protrude out from the body. That means they will emerge out from the body, from main body line. So that kind of structures are termed as appendages. Chordates have two pair of appendages, which can either be fins in case of fishes and limbs in case of terrestrial organisms. So one or both pairs may be secondarily lost in case of chordates. You can see that this whale that doesn't have hind limbs. Mud eel is a bony fish which belongs to order Anguilliformis. Okay, so it has four limbs. You can see here it has four limbs. These are visible here, but no hind limbs. And snake that are part of class Reptilia, they doesn't have four limbs or even hind limbs. Both are absent. In case of non-chordates, appendages are absent, and whenever these are present, there will be always more than two pairs. You can see here. So, class Insecta, it will be having three pairs of appendages. You can see three pair of legs, and scorpions, you can see. Okay, and in this praying mantis, you can see three pairs of legs. In arachnids, that means spiders, there are four pair of legs, and mites and grasshoppers and beetles, and in millipedes and in centipedes, you can see there are several pairs of legs. So whenever these appendages are present in case of non-chordates, that will be more than two pairs. Next is skeleton. Most chordates, they have living endoskeleton. Endoskeleton means which is present inside the body. Living, that means it will grow with the growth of body. So chordates do not have any need to shed the skeleton, as it will not hurt their development. And but in case of non-chordates, that will generally have exoskeleton. That means skeleton, hard covering, that is present outside the body. That can be horny, hard, calcareous, that means made up of calcium compounds, and chitinous. Chitinous means which is made up of chitin, and chitin is a mucopolysaccharide which is having a structure similar to that of cellulose somewhat. And so such kind of exoskeleton that should be periodically molted to allow growth. Otherwise the organism will not be able to grow its size. So it has to periodically molt the its exoskeleton. So you can see, so this is a chordate fish, bony fish. So it is the endoskeleton. And here is human being, mammal, that is also having endoskeleton which can grow with its growing years. Okay. And here is exoskeleton of this is a beetle which is termed as golden beetle, and this golden portion is its exoskeleton. And similarly you can see here exoskeleton of snail. Now next is coelom. Animals which have coelom are termed as coelomates. Coelom is present in most of the chordates and many non-chordates, but mode of coelom development is different in chordates and non-chordates, as chordates will be having enterocoelic coelom and non-chordates will be having schizocoelic coelom. So you can see here. Now this green structure, it is showing it is made up of endoderm, and in the center this is having archenteron, that means future gut, which will form the future gut alimentary canal. So this archenteron is lined by endoderm, and is this endoderm, it will grow outside in the form of pouches. You can see the emergence of pouches in the shape of a flower. So these pouches are, they will be separated off from this, they will pinch off from this, and they will grow into mesodermal pouch. You can see here that these pouches has pinched off. Now these are shown here in pink color, and they will be having in center a cavity which will be coelom, as coelom is bounded by mesoderm. So this pink structure is mesoderm and the cavity which is present here is coelom. So such kind of coelom, now it will grow further with time. You can see here this green structure is endoderm. This outer larger circular structure which is shown here in blue is ectoderm. Green is endoderm and outer blue is ectoderm. And this middle, which is pink, which is of pink color, is mesoderm. And the space which is bounded by this mesoderm is termed as coelom. Now this kind of coelom which has developed from the emergence of pouches from the enteron is termed as enterocoelic coelom, which is found in chordates. And the other kind is that there is mass of mesoderm and there will be splitting in this mass of mesoderm. So it will split from in between. Here you can see the split has arisen. Here the split has arose. Now it will grow in size with time and a coelomic cavity will be formed. So this will happen in case of non-chordates, and such type of formation of coelom is termed as schizocoelic coelom. You can see here schizocoelic coelom. So coelom in non-chordates and chordates is different because of its development method. Next is digestive tract. Chordates have complete digestive tract. That means it will be having two opening. Now you can see here that is digestive tract of some non-chordates, lower non-chordates, that will be having a single opening for inlet as well as outlet. So it will function both as a mouth as well as anus. But in case of chordates, there will be two separate openings and it will be tube within a tube plan. It will be having opening at both the ends. One will be mouth, then digestive tract, and on the posterior side will be anus, that will be used for ingestion of undigested waste. So see this organism, that is a platyhelminth, which is termed as the name of this organism is Dipsia, and it is having a common opening for food and for fecal waste. You can see. So here is a tube. Food will be taken inside with this tube, and the undigested remain. They will be expelled out. Waste will be expelled out through this tube only. And here is the digestive tract of any chordate. So that is of a cock. You can see here is the mouth, complete digestive tract, and on the other end is cloaca, or you can say anus. So digestive tract are different. Organisms above platyhelminth, non-chordate organisms, non-chordate phyla, which falls above the platyhelminths, they also have a complete digestive tract. That means Annelida onwards. Next is circulatory system. There is a considerable difference between circulatory system of chordates and non-chordates. Chordates, they always have a closed circulatory system in which blood flows in closed channels, which are termed as veins and arteries. But in case of non-chordates, only Annelida has closed circulatory system. All other has open blood flow. That blood will flow in open sinuses, which are termed as blood sinuses. Now, in case of chordates, heart will always be ventral in position. But when heart is present in non-chordates, it will always be in dorsal position. If you talk about the flow of blood, there are two kind of main vessels. One is dorsal, aorta, one is ventral aorta, ventral vessel or dorsal vessel. Now in case of chordates, the blood flow in dorsal vessel will be backward and forward in ventral vessel, but it will be reversed in the case of non-chordate. The blood flow will be forward in dorsal vessel and backward in ventral vessel. Chordates possess a respiratory pigment hemoglobin. Non-chordates, whenever the respiratory pigment is present, it can be hemoglobin, can be hemocyanin, or any other pigment. Hepatic portal system is present in chordates, but no such portal system is present in non-chordates. Now, what is this portal system? Hepatic portal system. That means blood collected from digestive system. It will be poured into the liver prior to sending it to the heart. So that kind of system is termed as portal system. When blood will be poured into an intermediate organ before it is supplied to the heart. Next is respiratory and excretory system. Now, lower chordates for respiration, they exhibit diffusion like urochordates and cephalochordates. But higher chordates, they possess gills and lungs for respiration, like fishes have gills, and terrestrial organism, they possess lungs for respiration and kidneys for excretion. But in case of non-chordates, there are special respiratory organs and special excretory organs like gill filaments and skin in Annelida for respiration, tracheae in insects, Ctenidia in mollusca for respiration, and for excretion there are present nephridia in Annelida and Malpighian tubules in insects and kidneys in mollusca. Here you can see in the next pic. Okay, so this is a chordate fish that possess gill for respiration. This is higher chordate human being, mammal, that possess lungs for respiration. Now, if you talk about non-chordates, so starting from annelids, so this organism that is polychaete, that is having gills. You can see these red structure, these are gills. So it possesses gills for respiration, and this organism belongs to arthropoda that has tracheae, which will open outside through spiracles, and internally it will be having air sacs. So there is extensive tubular network for respiration in the form of tracheae. So in arthropoda there are tracheae for respiration. If we talk about mollusca, so there is ctenidium, this red structure that is ctenidium. So that is responsible for respiration. Now, if we talk about excretion, there is proper excretory system comprising of kidneys and ureters and urethra and urinary bladder in case of most of the mammals. And if you talk about annelids, so there are nephridia. A different kind of nephridia are present, as you must have studied during the type study of earthworm, that there are pharyngeal nephridia, there are integumentary nephridia, and there are septal nephridia. So three kind of nephridia are present for excretion. Malpighian tubule in the case of insects, arthropods, okay, and there is kidney in the case of mollusk. Okay, that is shown here in this purple color, this one. So that is kidney of mollusca. Now we'll talk about endocrine glands. What are endocrine glands? These are ductless glands. That means they will be devoid of duct, and these will pour their secretion, their hormones into the blood. Endocrine glands are found in chordates as well as in non-chordates, but are much developed in chordates, much higher evolved in chordates, as you can see. So there is example of human being. So endocrine glands, main endocrine glands in case of human being is pineal gland, pituitary gland, which are situated in brain, thyroid gland, which is present in throat, and then adrenal gland, which is present just above the kidneys, pancreas is a heterocrine gland, so some portion of it function as endocrine gland and some portion of it function as exocrine gland. So and then ovaries in case of females and testes in case of males. So these are all example of endocrine glands. Now, in case of non-chordates, endocrine glands are found in arthropoda, primarily in arthropoda. So here you can see there is an example of scorpion. So here are two glands. One is androgenic gland. You can see here this dot just behind the testes. This is androgenic gland, and here in head, just below the brain, is present Y organ. That is also endocrine gland, and these secrete ecdysone, which control molting and reproduction in these organisms. Next are mesenteries. Now, what are mesenteries? Mesenteries are double fold of peritoneum through which viscera, that means visceral organs, are interconnected and are suspended from dorsal body wall. That means organs are hanged from the dorsal body wall, so that they must be kept in one place. They shouldn't move. So, and mesenteries are formed of mesoderm. Most of the chordates have mesenteries, but non-chordates, they lack mesenteries. Only two organisms among the non-chordates, they possess mesenteries for holding their alimentary canal. So now see the structure of mesentery. So this is alimentary canal, you can see. Okay, this is alimentary canal, and this veil like structure, this curtain like structure, this is mesentery. This yellowish orange color structure, this is mesentery. So that will hold this digestive tract in proper place in the body. So such kind of mesenteries are a common thing in case of chordates, but are lacking in non-chordates, except the two organisms. One is this Nereis, which is an annelid, okay, and other is Asterias, that means starfish, which is a echinoderm. Next is cleavage. Now, the cleavage pattern differs considerably in case of non-chordates and chordates. In chordates, there is always radial cleavage. Chordates and echinoderm, they possess radial cleavage, and all other non-chordate phyla, they have spiral cleavage. Now, what is the difference between these two? In radial cleavage, the first two divisions are similar in both the cases. So, first cell, it will divide by first cleavage into two cells. Second cleavage will divide this into four cells. So, first correlate of cells have been formed. Correlate means a group of cells having four cells. So, four cell group is correlate. Now, these four cells by third division, they will divide into 8 cells. Now, the second correlate will lie directly above on the top of first correlate. You can see the lower cell and the upper cell. So, upper cell is just above the lower one. So, such kind of division is termed as radial division, radial cleavage. And if the second correlate lies in the depression between the first correlate, that means where the two cells of the first correlate are joining, the upper tier will lie, the cell of the upper tier will lie at the junction of these two cells or depression of two cells, then it will be termed as spiral cleavage. Top cells are micromeres, that means it is not equal, division is not equal, not equipotential, while this radial cleavage is always equipotential. So top cells are micromeres and the bottom cells are macromeres in case of spiral division. So you can see the lower tier is having larger cell and the upper tier is having smaller cell. Okay, so if you see the top view, then it will look like this, the radial cleavage, that one cell is above the other, so you will only see the topmost layer, while in case of spiral cleavage all 8 cells are visible, as these are present on the junction of the lower tier, and upper tier cells are smaller. So you can see. So again you can see that radial cleavage is always equipotential, and it is present in case of chordates and echinoderms. So this is radial cleavage. So here are 4 cells and here are 8 cells. So this is side view and this is top view. So you can see here that each cell is situated exactly above the lower tier, and upper one is showing spiral cleavage. You can see that the upper tier is slightly twisted. It can be twisted towards the right side, so that will be dextral, or if it is twisted towards the left side, it will be sinistral. So you can see here it is dextral cleavage. because the cells are twisted towards the right. And such kind of cleavage is responsible for dextral and sinistral shells in apple snail. You must have studied that in your previous year. So, radial and equipotential cleavage is present in echinoderms and chordates, and spiral cleavage is present in non-chordates. Thank you. So, this was all about the similarity. Thank you so much.